Okay, I should note that we have this thing here that we call Lamont time, which is not, we're not quite at 7 o'clock Lamont time yet, but well, it looks like we can start anyway. So, this is great that we have, this kind of turnout on such a rainy evening. We really appreciate that everybody's come here. On behalf of the observatory, I welcome everybody to tonight's conversation. It's Seas of Change, the microscopic world that drives ocean health and climate. And I'll introduce myself. I'm Steve Goldstein. I'm the interim director of the observatory, and I'm also a professor of Earth and Environmental Sciences at Columbia and a member of the scientific staff here. And I'm also a neighbor. For those of you who live around here, I live in Spark Hill. So if you're living around here, we're neighbors. And I've been actually associated with Lamont in some way the first time, for a while. The first time I ever came here was when I was an undergraduate at Columbia, and it was in 1974. So it's been a while since the first time I came here. Okay, but I graduated, I left to go to graduate school somewhere else, and I had learned about Lamont, and it was just such an exciting place that I decided to take a master's degree and come back here. So I got my I came back and got my PhD here. The place where I was at, you might have heard of it, that I left. It's a place called Harvard. It's a little bit to the north of here. And then I graduated, and I left. I worked for a Max Planck Institute in Germany for 11 years, and I was offered a position here, and so I came here again in 1996, and I've been living in Spark Hill ever since 96. It's 28 years. Anyway, our public lecture series features talks by members of our scientific staff who are, you'll see, we're all passionate about studying our planet. Basically how it works, how it's changing, and what these changes mean for our society and our communities. We believe that the scientific understanding that we produce is essential to informing the solutions to the global issues that are confronting us. And tonight we're going to hear from Professor Sonya Dyhrman on her exciting and groundbreaking research in marine microbes. Sonya, like me, she's also a professor of Earth and Environmental Sciences and a member of our Biology and Paleoenvironment Division here at Lamont-Doherty. She got her undergraduate degree from Dartmouth and has a PhD from UC San Diego, from the Scripps Institution of Oceanography. She's highly recognized and is a fellow of the American Academy of Microbiology. Sonya is a marine biologist who studies how marine microbes interact with each other and their environment. She specializes in using genomic approaches, that is, through the DNA of the organisms, to study how their activities support ocean ecosystem health. Like other, we had hors d'oeuvres up there, and they're kind of coming back, sorry. Okay, like other Lamonters, her studies take her all over the world, from Antarctica to Long Island Sound. And her work combines field studies with laboratory experiments to query the vast network of microbes in the oceans and to forecast what those networks will look like in the future. And in addition to her research program, she's a devoted educator, both formally and informally. Besides mentoring and teaching, besides teaching and mentoring research at the university level, Sonya has developed outreach materials that have reached millions of children. And her work has been shared with policymakers at the UN climate meetings. These are the COP meetings. And I'm sure her lecture tonight will be illuminating and engaging. There will be time at the end of the presentation for questions, so please hold your comments until then. And with that, please join me in welcoming Sonya to the stage.
Thank you for that very nice welcome. And I extend the welcome to all of you. Thank you for being here tonight. And my not so secret goal here is to share the enthusiasm I have for the oceans, the microbial world, and hopefully leave you thinking about the ocean and microbes maybe a little differently at the end of the evening. Look at that big, blue, beautiful planet we live on. It's blue. That's the ocean. And we all know this, but the ocean covers most or the majority of the planet. And the ocean plays a profound role in our planet's functioning. In fact, I would argue that it's both the ocean and the microbes in it that make our planet habitable. So here's a picture of the ocean. I don't know what you think about when you see this picture. Maybe it's, I want seafood this weekend for dinner. Maybe it's like, oh, I wish a whale was like jumping out of the water, or a dolphin, or a seal. You think about the amazing marine ecosystems, the beaches that you visited, the boat trips that maybe you've taken. When I look at this picture, I think about those things, but I also think about the unseen world. And that is the microbes, which you have to envision might look something like this cartoon. We can't see them with the naked eye, but they're out there living in this vast network where they interact with each other, they interact with their environment. We have different species of microbes. Some are feeding each other, some are eating each other. Every day, some are getting infected with viruses and dying. They're growing. In fact, about 10 years ago, my colleague Ben and I discovered that these microbes were even talking to each other. They have a chemical language. They make these signaling molecules. Different microbes have different languages. And we're now starting to literally try to listen in on the conversations that these microbes were having. We went on our research cruise and added signaling molecules to samples and tried to look at what happened. It was sort of like screaming, hello, microbes, and trying to figure out what happened next. Not only are these microbes kind of amazing and all those intricacies and interactions, but they carry a global level impact. One of those is in the carbon cycle. And so just to kind of give you the punchline here, those microbes in the ocean process about 50 petagrams of carbon every year. Petagram is hard to conceptualize, these big, big numbers, but that's about 50 billion, with a B, metric tons of carbon. The microbes. They can do that because they are incredibly numerous. So here's a picture of microbes, all of those green and red dots. imaged and stained so we can look at them with the microscope from 1 microliter of seawater in the surface ocean. That's the size of that little drop on my friend Grieg's finger in this picture. It is literally a pinhead-sized drop of seawater, and look at all those microbes. There are about 1,000 microbes in every pinhead-sized drop of seawater. And you think about the size of the ocean, that's pretty incredible. I don't know how well you can see them from the back, but there's a faint lawn of really smaller green dots. Those are actually viruses in this sample. They're viruses that infect microbes, and there are about 10,000 viruses in every pinhead-sized drop of seawater. Now, if we were to take all of the microbes, just to, again, try to conceptualize these numbers, and put them in a box with no room, so all the microbes in the ocean, and we shove them into the box, there's no other room, we have this size of box, and if we took every other animal in the ocean, so all the whales, all the sharks, all the starfish, all the fish, all the clams, and we cram those guys into a box, no room, do you know which box is bigger? The microbe box. It's actually twice as big as the animal box. That's pretty incredible. Now, most of the microbes just look like little circles, but I want to show you a few of my favorites that are a little bit larger, because I have some really nice pictures. Here are 3 different microbes that come from the surface ocean, and I'm actually growing them all in my lab just across the campus here. So they're personal favorites as well, and we do experiments on them. So the one on the far left is a calcifying microbe. It biomineralizes and makes those chalk plates that it covers its cell with. That type of microbe is very important in the carbon cycle because it's got those hard parts on it, and that means it wants to sink. So it's going to sink its carbon out of the surface ocean. Interestingly, it's also why the White Cliffs of Dover are white. It's very numerous, it's been around a long time, and it makes those chalk plates. So you can even see blooms of this organism from outer space. The middle one is a diatom. It also biomineralizes, but instead of a chalk cell plates, it makes a glass cell wall. So it strips silicate out of the water and makes shells out of silica. It also is hard and heavy, and so it also wants to sink. Diatoms tend to be numerous in coastal waters. They really like nutrient-rich environments. And this last one is called trichodesmium. We call it tricho for short, and it's actually very small cells that in this image are all connected into filaments, and then those filaments are organized into these 2 different colony morphologies. So this is a whole colony of microbes. One of the cool features about that particular group is that they can fix atmospheric nitrogen. So they can take dinitrogen gas, which is a very hard bond to break, and they can break that bond biochemically and then make bioavailable nitrogen not only for themselves, but for other microbes in the system. So it's a way of injecting what we call new nitrogen into nitrogen-poor regions of the ocean. The other big facet of all of these microbes is that they're phytoplankton. Phyto for photosynthesis, and plankton meaning they drift with the currents. And so they are photosynthetic, which means they use energy from the sun, of course. They take CO2 and they make sugars and other cellular carbon building blocks. This invention came about 3 billion years or so ago, and it literally changed our planet and oxygenated the atmosphere, because that's one of the byproducts of this reaction. Because they're phytoplankton and they're photosynthetic, they carry pigments for harvesting light, just like land plants. They have chlorophyll, and we can get a sense for how globally significant these organisms are by imaging chlorophyll from satellites, and that's what I'm going to show you in the next movie. So all the color is chlorophyll in the ocean. So here the globe is going to rotate around, and every time you see the warmer colors, the reds and greens, that's an increase in the amount of chlorophyll in that system. And what you're seeing is chlorophyll increasing every time nutrient-rich seawater is coming into the surface ocean. You can see the Amazon plume there rotating around, lots of nutrients in the Amazon River. You can see that in the coastal zone where nutrient-rich water is being brought to the surface, and along the equator where nutrient-rich deep water is also coming into the surface. So amazing global reach for these cells. Now, if we were to drill down a little bit and think about those microbes and take a cross-section of the ocean, I want to first talk about food web dynamics. These phytoplankton underpin marine food webs. So they use light energy to fix that carbon into biomass, and then that carbon gets transferred through the food web. Small phytoplankton get eaten by microzooplankton and zooplankton, which are just little like animals that also drift with the currents. Those can get eaten by fish. You could have fish eaten by sharks, and so on. And those food webs support all of global fisheries. They also support the food webs that bring about penguins and polar bears and whales and, again, sharks, even shellfish. All of those, what we call in microbiology, charismatic megafauna, start with phytoplankton in the ocean. They're churning away, using that light, fixing carbon, and then supporting that global ocean food webs. Now, we can also think about this cross-section in terms of carbon. So we know they're fixing carbon into biomass, and that carbon is going to move around food webs, and some of that carbon is going to get respired and go back out into the atmosphere. But some of that carbon, maybe because it's locked up in one of those heavy phytoplankton, is going to sink out from the surface water. And if it makes it to the deep ocean, there, it will have about 1,000-year residence time. If it makes it to the sediments as a hard part, then it's essentially locked away forever. And in fact, this biological pumping of carbon through the system and to the deep ocean, we call the biological pump. Again, now we can look at another map and try to compare this with what's happening on land. So we're used to understanding that the Amazon rainforest and other systems are really important in terms of trapping carbon and moving it into the system. But the ocean microbes account for the other half. So every tree, every blade of grass, every savanna, all of that photosynthetic carbon fixation is only half of the global story. The other half happens with microbes in the surface ocean. One of the neat things about this NASA animation, where you're seeing again patterns of photosynthetic biomass, is you can see the seasons going back and forth. And if you've noticed, that's happening in the ocean as well as on land. So there are seasons for these phytoplankton in the ocean. And when I watch this movie, I think about the flow of carbon and the flow of oxygen, and it's almost like the planet is breathing. In fact, when we get to the CO2 record, which I'm going to show you in a little bit, you can actually see the ocean in that record and the land moving that basically makes a little squiggle in the CO2 record of the atmosphere. So there's a vast network of microbes driving ocean ecosystems and the health of those ecosystems and climate, moving a lot of carbon. But how do we observe the unseen? Now, we can go and we can look at samples with a microscope, for example, and observe what cells are there, but a lot of them look the same. Even with the most high-powered microscope, there are bacteria and other microbes that you can't tell apart. So that's limited, and we need other tools to try to track who's there and what they're doing. And this is when I can tell you it's a really exciting time to be a microbiologist. We have tools now that I didn't even dream of when I was in grad school. And that's in part because we've been able to capitalize on the genomic revolution that came about largely through innovations that came through the Human Genome Project. So just like we can all appreciate, sequencing the human genome will hopefully is telling us a lot about who we are, what our blueprint is for being human, can help us diagnose and understand diseases and what might be coming. And that human genome sequence drove down the cost of sequencing DNA, and it also led to lots of innovations and new sequencing technologies that we're able to capitalize on to basically do ocean microbe genomics. This is the cover of Time in 2012. And again, it's asking, want to know my future? We can tell things from the human genome, like personalized medicine, how to tell maybe what diseases you might be getting and how healthy you are. And we're essentially trying to do the same thing, but for those ocean microbes and ocean samples. And in fact, just the year before this Time cover, I was on a team that sequenced one of the very first phytoplankton genomes. This was actually for a harmful species of phytoplankton called Oreococcus. Now, it pains me a little bit to talk about a harmful one because the vast majority of microbes are beneficial. They serve important roles for our planet. But there are a few that make toxins, or in this case for Oreococcus, are so good at growing that they actually grow to really high densities and choke out the ecosystem. In fact, we call them ecosystem destructive. This organism blooms and causes problems in the mid-Atlantic. And in fact, the isolate that we sequenced came from Great South Bay and Long Island. And what we learned from looking at the genome sequence was that this organism was uniquely adapted to living in urban impacted waterways. It had all kinds of metabolic capacity for dealing with metals that are often toxic to other phytoplankton. It had lots of special proteins for working on light harvesting, for growing in shady environments where there's lots of cells. And so this was the first exciting pass in being able to use genomes to understand more about these organisms. I want to just give one more example. And here we have, again, another calcifier. And this paper and study came out just a year after that Time cover in 2013. And this was the first time we actually sequenced multiple genomes of the same organism. And what we found was that, in fact, there's a core set of genes, which are just the organizational units and genomes, that make this organism, which I call affectionately EHOX, EHOX. But then each strain had a set of variable genes. These genes are only found in certain strains, color-coded here. And we could look at that variable component, the genes that aren't the same across all those strains, and use that to try to predict and understand where we find certain strains in the environment, and if that genetic capacity is helping to determine where we find certain microbes. Just to show you how everything has continued to advance in 2013, it took us about 10 years with funding from the Department of Energy to sequence the genomes that went into this original study. And I currently have a project where we're sequencing 15 more genomes, but it only takes us like a year. So we can do it much faster and with better technology and for less money, which is pretty incredible. And this is giving us this first blueprint into who these guys are and what they're capable of doing. And it also growing many more tools that we can then develop and use to analyze these data. In fact, we've moved from the bottleneck being sequencing to the bottleneck being, what do we do with all of this data? There's a storage issue, and there's an issue around even analyzing the data, which it now takes. I can't analyze it on my laptop anymore. It takes a high performance computer cluster and someone who can write code to talk to that system to analyze these data. And in fact, now it has really retooled how I think about training my own graduate students, because they need that background in computational work. And it's important for me to be able to build capacity for that nationally and globally. And so I'm actually a part of a group that runs workshops on genomics now every year for international capacity building in these computational genomics approaches, so that we can have more environmental scientists coming into the program and thinking about how to use these tools moving forward. So when we think about visualizing the unseen microbial network now, we're actually capitalizing on more than just genomes. And here what I show is just the biological cascade that happens in cells as they interact with their environment. So we have the genome, which is like the blueprint. It has genes based on signals to that organism. It will essentially turn on or transcribe those genes into RNA. That RNA can get made into proteins. Those proteins do the work of the cell in building out the chemicals and building blocks that hold the carbon. And by visualizing different points in this biological cascade, in particular, we focus a lot on the RNA, which we can sequence. We can go out and ask from the RNA signal, who's there and what genes have you turned on? And that tells us something about their cell's activity. For example, here's the output of a set of experiments that my former postdoc Gwen, you can see right there, and former Barnard undergrad Liv Williamson did on phytoplankton, where we grew them in the lab under different conditions, including a control, high CO2. We also starved them for phosphorus, starved them for nitrogen, and compared them to control conditions. We sequenced the RNA, and what you see here is the RNA signal color-coded across different parts of that cell's metabolism. And you don't need to worry about the details, but if you look, you can see each of these sets of bars doesn't look the same. There's a different fingerprint for each of those conditions. And this is one way we can visualize what these cells are doing. We can do experiments in the laboratory and then understand how these signals are modulated for interpreting them in field populations. So a lot of what I do is to go out and also take samples in the field. And maybe in terms of thinking about this, it's less about a sea of microbes and more about a sea of genes, where we can go out, generate this sequence information, and again, ask and answer questions that I only dreamed of. It's such an exciting time. And we ask and answer these questions like who's there and what are you doing? Are you talking to each other? How much carbon are you using? Across a whole variety of different environments. This image I chose because I don't know if anyone can recognize it. This is Great South Bay in Long Island, where that harmful alga came from. And in fact, you can see the blue ocean water there. There's a brown tide happening when this photo was taken. And we've been using those genomic signals to try to understand the factors that drive these blooms, why they occur, and how we might mitigate them. I also chose this photo mainly because Antarctica is amazing and really special. And if you have any questions on what it's like to get to be there afterwards, I'd be happy to answer them. This picture I took, it was actually Christmas Day. We had been on the ship for about 4 weeks, and we had special permission to take a Zodiac to the continent and walk around. There was a big penguin colony there, and so I got to take a picture of the ship. This is the research vessel, the Palmer, which is, of course, an icebreaker. Now, as I said, it's a really exciting time. And we're able to track these microbes in ways that I didn't believe we could. But it's also a really pressing time to try to ask and answer these questions. As we know, the ocean and the climate are changing. And so we're trying to understand these systems while the baseline is shifting. There are plastics, both what you can see here, and dangerously the ones that you can't see, the really small plastics. We have increasing temperatures. We have increasing nitrogen, phosphorus, other resources. And of course, we have increasing atmospheric CO2. Now, the ocean is responding to these changes. And in fact, I'm coming back to this CO2 record now. You remember me talking about the squiggle when we were looking at that movie of the planet breathing? This squiggle that you see in the atmospheric CO2 record is photosynthetic biomass seasonally driving that squiggle. But overall, of course, CO2 is going up. And if we look at what the ocean is doing, right near the Mauna Loa Observatory where they're taking these measurements, you can see ocean CO2 is going up, and this drives ocean pH down. This change in pH changes the carbon chemistry, among other factors. And so again, the baseline is already changing. And the number one question that I always get, and frankly, the primary question driving my research efforts, is how are these ocean ecosystems going to change in the future? How do we sort that out? The short answer is we don't know exactly how they're going to change in the future. And part of the challenge here is that we don't fully yet know the rules in which the system is operating. So what I'm trying to do with my research is I keep telling my students, we have to understand the rules. Then we can predict what's going to happen. And we need to know how sensitive this microbe network is to changes in the environment. We approach this with a lot of different tools, not just the genomic ones. We use models. We actually use these models to try to understand what don't we know, what are we missing in the network. We do field studies to observe the network and try to learn the rules. And we do laboratory experiments to test the sensitivities. So what I want to do for the last few minutes of the talk is actually take you through a field study that we did and show you some of the data that we're looking at and thinking about as we try to address these questions. One of the places where I do a lot of research is actually called Station Aloha because it's right near Hawaii. It is in an area of the ocean that we call the North Pacific Subtropical Gyre, or the NPSG for short. I've been working there in that system, doing cruises almost every year for close to 20 years now, a lot of my career. It's a pretty incredible opportunity. And this is one of the areas of ocean sampling where the National Science Foundation funds a time series. So every month they take a ship out to this location in the ocean and take a series of samples, and they've been doing that for decades. And so when we do research in this spot, it allows us to put this longer time series set of observations around it. So the research expedition I want to talk to you about tonight, I like to call the Mahalo expedition, mainly because oceanographers like to name their research cruises. And we talk about this as the Mahalo expedition because Mahalo is thank you in Hawaiian. And this research cruise was really special because we had not one, but 2 ships of scientists out in this spot trying to take samples for several weeks. And this was supported in a collaboration funded by the Simons Foundation, and we were very grateful because it's almost unprecedented to have 2 research vessels in the same spot taking samples. Now, this is a picture of one of those ships. In fact, I'm on there somewhere. This was taken from the other ship during the cruise. RV stands for research vessel. This is the Kila Moana. It is operated out of Hawaii. As you may or may not know, Columbia operates a ship in the U.S. research fleet. It's called the Langseth. And I actually haven't sailed on the Langseth. I've sailed on many of the ships in the fleet, but we go to wherever the ship is that's closest to the site that we want to study. We don't go with the ship operated by our own institution. We go with whichever's closest to the environment. These research vessels are amazing tools, and it's a great privilege to be able to go out and get these samples. And on this cruise, we ship all of our gear, and there's a lot of it, to Hawaii. And then we load it all onto the ship. And I took a picture from the dock. Now, if you can't see the research vessel, it's that one. I love going to sea. I calculated it once, and this was a while back, so I've spent over a year of my life, if you add them all up, on a research ship in the ocean. And but I have to say, even though I love it, it is slightly demoralizing loading your research ship that you're going to be on for 5 weeks next to a cruise ship. But anyway, it's a special privilege. And this Mahalo expedition, one of the things that we were doing is trying to look at the carbon cycle. And the way we were looking at it is with signaling from light. So we were basically looking at day-night samples taken as we tracked this water mass with a drifter off the coast of Hawaii. So here's Hawaii, the Hawaiian Islands. Here's Station Aloha. Because of the physics at the time, we sampled a couple of degrees north of Station Aloha. We put a drifter in the water, and we figured, every day the sun's coming up, that energy is driving that flux of carbon dioxide into the water. Can we start to bring a bunch of tools to bear and see what we can learn about the fate of that carbon and how it might pass through the microbes. So we sampled every 4 hours for 6 days, and then we repeated it all again. So it was very tiring. Here's a picture of us sampling for the water. So we have these instrument packages with bottles that we send down and then collect water at different depths, bring it back onto the surface. Here's a former Lamont grad student, Kyle Frischkorn, helping to move the water into the lab. I'm there in the background, frankly looking pretty tired. Although in fairness, I was sampling on the 2 a.m. time point shift, so I'd probably been up all night at that point. And then we filter the water to collect the microbes. It's challenging. We have to do it gently, but quickly, because the RNA signals that we're trying to study can turn over and change in minutes to hours. And we had to do all of this in the dark. So we actually used red light in the laboratory to process the samples. We were really worried about light contaminating and changing our signals and whether this was even really going to work, frankly. So what we were tracking there are the RNA and the metabolites or biochemicals. So we were looking at this point in the cascade for signals. And we took those filters, extracted the RNA, sequenced it, and looked at the signals. Now, before I show you the data, I want to say that, again, this is amazing technology, and we generated over 100 million sequences for every single sample that we took. This is an incredible data set, and it is a big data puzzle, sort of like a 100 million piece puzzle and then some. So our first question was, can we really track patterns of carbon flow this way from the RNA signals? What can the microbes tell us? Here's part of the answer. So in blue are RNA signals that are turning on photosynthesis, in red, turning on carbon fixation, and in brown, turning on the TCA cycle, which relates to carbon processing and those building blocks in the cell. And it still amazes me almost that we can see this. Look at those beautiful day-night patterns. This is from one organism, our friend E. hux. And you think about what we had to do here. We had to fish for the RNA that belonged to this organism, hope that we didn't light contaminate it when we took the samples, and then process 100 million piece puzzles and generate these patterns. And you can see the dark bars are nighttime and the whiter bars are daytime. And even before the sun comes out, these cells start to make their photosynthetic pigments and get all their machinery ready. And then the sun comes up, and they are like, oh, they did their job. They harvest that light. They pull down the CO2, and you can see that happening in these pathways. And then they start to turn off that aspect of metabolism because the carbon is moving on through the cell into the TCA cycle, which comes up later. So if you stop and think about it, again, this is visualizing the flow of that carbon through this one organism. I want to just briefly highlight that this is work done by a Columbia Bridge to PhD fellow from my laboratory, Maria Hernandez-Lamont. This is a program that brings post-baccalaureate students in for research for 2 years and tries to bridge them into PhD programs. This data set was really admirably analyzed by Maria. She did an incredible job. This was her very first scientific research paper, and now she's a PhD student in the geosciences at the University of Chicago. So really amazing. Thank you, Maria. So before we end, I want to take you through one more data set from this cruise. And that is looking, instead of just at one organism, at the whole community. So briefly, here in the top panel, again, we have day-night patterns. This is particulate organic carbon. So this is just the carbon pool locked up in cells. And what's pretty remarkable about this system is, again, just by tracking the bulk carbon signal in the particulate matter, you can see cells are growing through the day. They hit the evening and they start to die. They're getting attacked by viruses. They just run their life. And then every day they grow up and then they die. They grow up and then they die. We can look at the carbon molecules in the cells. Two of the big storage molecules for carbon, that's shown here in these lines. And you can see they mimic that bulk pattern. So again, these cells are making these organic carbon compounds as they grow up. And then those molecules are lost to the seawater as they die. So then we can look at the transcripts. And first, let's just focus on the pink line, which is photosynthesis. And here we combined it with carbon fixation. So again, you see the phytoplankton growing up, photosynthesizing, moving all of that carbon into the TCA cycle, shown in green, and building up that carbon pool. And then that carbon is released. That carbon is in the form of things like sugars. And so we can actually look at other microbes and ask who is transporting sugar out of seawater. And that's shown here by this yellow line. So these are microbes that are turning on their sugar transporters to bring those organic carbon compounds into the cells. A couple of cool features about this yellow line. These are, you can see that it goes up and down with day-night cycles. These microbes are not photosynthetic. They don't have any pigments. They cannot tell that the sun is out or not. They have no way of knowing. But they are completely tuned in to that flux of organic carbon that's going to come through the system every day. They know it's coming. They turn on their sugar transporters, and again, we're visualizing that pathway of carbon through the microbes. So I think this is really exciting progress in understanding the microbial network and its role in ocean health and climate, tracking that carbon flow. And there are still uncertainties, but I'm excited to report that we have a new program, some colleagues and I, where we're trying to move the needle on really further addressing those uncertainties. I talked about this new work in terms of addressing carbon cycle uncertainties at the COP27 meeting that was held. This was the COP 2 years ago. And one of the exciting things about this particular climate meeting was that the ocean was finally represented with national stakeholders at the meeting. And I participated in programming in the Ocean Pavilion, talking about marine microbes and their amazing role that they serve in terms of ocean health and climate. Before I wrap up, I just want to thank my Dyhrman Lab family, recent past and present, the many different sources of funding that have supported this work, and just hope that I'm leaving you with, the time is now. There is this amazing confluence of need and opportunity to more fully resolve the rules which govern microbial networks in the ocean and their role in ocean health and climate. And collectively, I would say this knowledge will help us better understand marine ecosystem resiliency on a changing planet. I'll just finish by saying, hopefully I'm also leaving you with, microbes are amazing. Thank you. Yes.
Do you think someday you'll be able to use your research to influence microbes to actually process more carbon, or is that a dangerous thing?
Well, let's see. There are already efforts underway to figure out how do we get microbes to help us pull carbon out of the atmosphere. There's kind of different flavors of how we might do this. Some people are looking at engineering microbes. In fact, one of my former grad students works in algal biotech, where they're trying to understand how do we do that sort of thing. Another flavor is to grow up algae on land and use that to pull down carbon. And then you have to essentially bury or sequester the algae. Phytoplankton. And there's also a lot of ongoing research in what we call marine CDR. And what I talked about at the COP panel, and CDR, as I should have said, is carbon dioxide removal. So marine-based CDR approaches, almost all of them intersect with microbes in some capacity. There are some actual strategies that are being thought through where we try to just fertilize the ocean and grow more microbes and photosynthetic microbes and get them to fix the carbon. It's hard to predict whether you're going to get the guys that really want to sink, and so there are challenges there in terms of basic research understanding. And what we talked about at the COP27 meeting was how do we work as thought partners in thinking about carbon dioxide removal and the basic research to try to bring them together to move that forward as quickly as we can. Yeah.
So with the changes in the atmosphere over the last several decades, have you noticed any pattern changes in the oceans or in particular locations that you've been doing research?
Well, so yes, there are regions where we see the microbes already changing. In many cases, there are multiple environmental changes happening at once, so I'm not sure I would feel confident saying it's for sure CO2. But yes, we are seeing changes in microbial communities. The very few places where we can see those changes happening, though, are at places like the time series, where they have samples over a long enough period of time that we know we're not just seeing natural variability in the system, which is also very high. It's very hard to predict right now. If I go out, I can tell you in general the kind of microbes that are there, but specifically who's going to be there and what they're doing. We simply don't have that kind of predictive power because we don't understand the rules. But for example, there are long-term monitoring programs in the Southern Ocean off the coast of Antarctica, and there they are seeing changes that they think are climate-related in the microbial communities. So I guess I'm wondering what your question is.
Well, I'm thanking you for telling me why he was arrested.
Oh, okay. There have been a whole series of experiments historically where we added resources to the ocean. This was mainly done around iron and trying to understand the role of iron in driving phytoplankton growth. But those kinds of large-scale experiments aren't even allowable right now.
We were talking earlier about going in and tweaking microbes so that it improves sequestration of carbon. But you also said that the whole microbe community is the basis of the entire ocean environment. How do you prevent unwanted consequences of going in and tweaking one community, and how do you prevent unwanted?
That's a really important question, and I want to make sure that I'm clear that when we talk about engineering microbes, it would be in a farmed context or in a contained facility. No one is advocating engineering phytoplankton or other microbes and putting them back into the ocean, because we would not be able to control what happens. And there would be a lot of uncertainty around that. There's a lot of uncertainty of even some of the ideas around CDR approaches where we're adding or fertilizing the water, again, because we don't want a harmful species to grow up, for example. But thank you. It's an important question. There are actually, we can't even take, and we don't take algae cultures on the ships with us, for example, because we do not want to risk introducing something to the environment. Yeah. Cool.
Oh, so you showed us that the ocean pH is dropping. Is that affecting the calcifier's reproduction and, in turn, atmospheric oxygen?
Well, thank you so much for that question. So you've hit on an important point. Those calcifiers that I study are just like corals. You know, they're made of chalk, and if you change the pH, they do not biomineralize as well. Many of these strains, however, will grow just fine at those pHs, and they can grow just fine even if they're not biomineralizing as well. So it may change their fate. They will grow fine, but you are right, it would potentially change their fate if they do not sink as much because they're not carrying as much calcium carbonate. That's an active area of research. Thank you for that question. Maybe one more question. Yeah.
Do you see any immediate possibility of commercialization of your research? I mean, is there any? I know it seems you're afraid to do too much because you're not sure of the consequences. But do you see anything coming that will be economically advantageous to use your research?
Well, I think that in a sense, perhaps the basic research that I do in my lab, and that is common here at Lamont, again, informs some of those approaches. So every time we sequence the genome and we learn more about the blueprint, that offers an opportunity for both further study, but also maybe for engineering microbes to do different things, again, in a contained capacity. Also, everything we learn about field populations, again, could be related to and used to inform future carbon dioxide removal plans. Those carbon dioxide removal plans, there are many, many companies really investigating and looking into that. So in that sense, it is a commercial opportunity.
Okay,
Thank you.
Thank you very much, Sonya. So, I mean, there was a question about climate change and impacts. It turns out that I teaching a course in field geology where we are going to Barbados over spring break. So this is spring break over in Barbados, right? But they'll be really happy when I say, okay, be ready at 7:30 tomorrow morning to get in the buses so that we can get out and look at the rocks. Anyway, I've been working there for the last 25 years doing research, and really over the past around decade, suddenly we've been inundated with macroalgae, okay, which is sargassum that's basically growing in the Atlantic and inundating the beaches. And it has really caused a lot of problems in Barbados and other islands. So we see that. I see that, and I'm not a marine microbiologist or a marine biologist by any means. In any case, thank you so much, Sonya, for sharing your cutting-edge work here. And I'd like to thank the audience for joining us this evening. We got a great crowd tonight. We're grateful for your interest in this profoundly important work. We look forward to continuing this. I'd like to invite you all to join us at our next public lecture. It's going to be it's going to be on April 24th, so it's right around Earth Day. The lecture is going to be called Clearing the Air, Understanding New York City's Air Quality and Climate, and it will be given by Roisin Comain, who's an associate professor of Earth and Environmental Sciences here, and a member of our Ocean and Climate Physics Division. So I just want to note that within each year, we gain new insights into our planet, but there's a long way to go. And it is the next insight on the horizon that keeps our researchers at Lamont excited to learn more about how the Earth works and how it changes. You can help advance our scientific discovery. We do take donations, and we appreciate every donation that we get. So we thank you for coming here, and I would just want to say, please consider supporting us, and thank you very much, and good evening.